Hi, welcome to the last session today for InnoFX Live. We're here with Les Edwards from SKAN. SKAN or SKAN?
Either way you like to pronounce it is great.
Okay, good.
It sounds more European when you say SKAN.
Yeah, well then that has much more uptake.
Yeah.
It got to sound European. So, one of the things that I got to tell you, a lot of times, there was over time, developmental challenges become more and more. There's always hurdles, and I think one of the things that's become a showstopper for a lot of folks is all of a sudden they got this terrific compound and thing shows up as potent. So the answer really comes back to, so Les, I mean, when you talk to, what is somebody looking for? I mean, what's the first thing they do when they find out? Are they going to be in that danger of being in that situation? So what do they look at?
First thing is defining what potent means. There's a lot of different bands of potency. A lot of people initial reaction is fear. Second is, what is the solutions out there? And the third is trying to figure out what solution makes sense for me. So for example, if I have an injectable and it's a chemotherapeutic and it is injected into the body, it makes you think, wait a second, how potent can this thing be if I'm going to inject it into somebody's veins? Well, potent doesn't mean just that it's potent at the delivered dose type of concentration. It's also how you're handling it in the interim. So you have to look at all the different stages of manufacturing, decide what is potent, at what stage, and how to handle it.
Okay, so we got that part. So I noticed you mentioned aseptic. So are aseptic potent compounds any different than aseptic? I mean, what's the difference there?
So aseptic materials are typically those that need to be manufactured and all the different sterile parts need to be put together. So they cannot be terminally sterilized or sterilized at the end of the process. So aseptic needs to be all put together. A lot of new biologics and a lot of potent compounds end up being aseptic compounds. And it adds a whole nother layer of complexity, because not only do you need to tie together all those different components, but you need to now consider safety as well. When you think aseptic, you're always thinking, I want to protect the product, I want to protect the product. Now, as soon as you start bringing potent in there, your primary concern is, I want to protect my operators. And now you have to balance that.
So, give us what would be an example?
Okay.
We got an aseptic potent as opposed to a regular aseptic. What?
Okay. Let's say that you have again, a chemotherapeutic product, and you want to manufacture it, say, in an open, standard aseptic processing environment. You have airflow going over there where you have the liquid gets exposed to aerosolization and things like that, and that air now gets exchanged into the surrounding filling environment. And now, all of a sudden, you have safety issues, because in regular aseptic, you didn't worry about it because it was just a residual of a product that's very dilute. It's not a problem for the operators. But now, all of a sudden, you have a potent compound, you need to protect the operators. What are you going to do? So there's a whole range of solutions that you can go into, from adding, say, personal air protection devices for the operators and just continue operating like you did before. Or you have to maybe do more extensive personnel protection, where you might have full gowning and full respirators and things like that, to other solutions, which I personally work with quite a bit, was isolation technology, where you actually build a box around your process to contain it within the system.
So the isolator is essentially just it doesn't become part of the process, but it becomes sort of like a stumbling block to run the process.
In a way.
I mean, in other words, it's just another thing you got to work around it.
Absolutely, yeah. It's a barrier, and barrier certainly implies more challenges, right? So all the different processes you need to look at, from getting your equipment and processes and product in and out of the isolator, is just as challenging as doing the process itself inside the isolator with a barrier in the way. So if you're working through glove ports now, you have less ergonomic access. If you need to make adjustments to machinery or equipment, that becomes more complex. So also there's all the questions of how you validate it and how you need to run your operation, whether you run it under positive pressure, negative pressure, all those type of considerations need to be
So where does
taken.
the isolator get in? Is it upstream, downstream, filling, processing? Where do you put this, where do you apply this?
All right, so one of the first questions you asked me was, what do I do? And I was saying you need to evaluate your process. So maybe your whole problem doesn't have anything to do with filling, and you can do your final fill finish of a dilute product in a standard aseptic operation and not have to do any additional safety aspects. But in your upstream compounding area, you have this powder that needs to be added to a tank, for example. And you can't do that in an open environment without having some serious safety implications. So what do you do there? Maybe you build an isolator around your tank, and then you transfer your powder into the isolator beforehand. You still have to do it in an aseptic manner. Okay, so there's various solutions out there with different levels of containment. You bring that in, and then you need to open up your powder, and you can dump it in there without having the powder plume up or have any problems there. Then you close that off. Maybe you do some sort of mixing step. Maybe you do a transfer of that, and maybe it's safe enough at this point, or it has been reduced from a higher band level to a lower band level where you can do fewer safety type measures.
And it really comes down to not just the potency of the compound as a dose, but the concentration that you're dealing with. In other words, if you got 5 kilos of pure drug substance as opposed to a 10% solution, does that change the band that it sits in?
It absolutely can. Sometimes it doesn't, but usually it does, yes. And again, when you're dealing with injectables in particular, you're looking at something that, when you expose a patient to it, it has to be at obviously a safe level. All chemotherapeutics work by the principle of it's kind of like poison, right? So you need to do a controlled poisoning of the patient, hopefully preferentially poisoning the cancer cells, and then giving the patient a chance to recover and heal.
So, and how do you evaluate that, Lester? You go to the site? How do your clients work with you on stuff like that?
So typically, you have some sort of basic information about the substance. A lot of new drugs, you don't, and you kind of treat those as the most dangerous methods, and you use the most conservative manufacturing technologies. But that's one of the big challenges in any sort of major aseptic processing. Once you've been through the clinical phases and you're starting to get into commercial production, usually the product is pretty well understood, and these levels have been established, and they're considered bands. SafeBridge has a scale that's well accepted by the industry, and whether it's a band 1, band 2, band 3, there's certain prescribed occupational exposure limits, and then you in those ranges of occupational exposure limits, there's certain prescriptive requirements for your manufacturing solutions.
So it's possible that in a lot of cases your clients come to you before they've done any sort of toxicity study?
In my former life, yes.
Right.
I used to do contract manufacturing and aseptic handling, where I had a laboratory there where we would need to do some testing with the customer and do characterization of the product. Okay, but usually the toxicity studies are all done before any clinical studies are performed, and these are done, through chemical analysis and also through animal studies and the like to understand the lethality of the
Right. Yeah, there's
substance.
certain modeling that you can do and make certain assumptions based on basic structure, design, and
Exactly.
yeah. Okay.
Right.
And a lot of, do you find that a lot of people they do a risk assessment simply based on things like solubility, for example?
That is one factor for sure. The other is, the manner of administration, and also, how the patient is going to end up being exposed to it, and or how the operator is going to end up being exposed to it. One of the other big considerations there too is, okay, I've done my aseptic process, I've protected everybody. Now I need to, let's go back to the compounding example I gave. We put our tank inside of an isolator, great solution, but we had this plume of powder that got all over the outside of the tank when we did it. Everything inside the tank is safe and pure and great, and we've contained that, but now all of a sudden we need to get rid of what was on the outside of the tank and the residue. So you end up having manufacturing challenges in cleaning and changeover. The next leading question there is, well, what is clean? So going back to some of those lab studies we were talking about, when you're characterizing a product, you understand its structure, but you also have to know how to inactivate that product. If it's a biologic agent, maybe it's as simple as running some sort of biodecontamination process. If it is a potent compound that has some sort of chemical in it that can be inactivated, then you add another chemical to it, it neutralizes the compound, and then it can be safely removed and rinsed from the process. Also, what level is safe or not safe? So you have to do these cleaning assays and determine, to what level of detectability is it still safe. Some people use simple methods like a total organic, TOC or total organic carbon, but that may be non-specific. So that's great when you're trying to figure this out and trying to figure out the specific compound, but it's best to have assays around your specific compound to know that you've been able to clean to the point where it's clean enough to be safe. And then a lot of times people do two-stage cleaning where they get it clean enough to be opened up, where the operators can get in there and do a thorough cleaning. And then their next level is, well, now the operators got in there, I don't want any cross-contamination, or I want to be able to introduce another product. I can't have any detectable levels in there, so I have to do another, I call it a GMP cleaning, after the safety cleaning. And that's really two stages that are really important to understand, so not only how to do them, but how to validate them.
So it sounds to me like you have to have primary and secondary type of strategies here. So how does this work?
Absolutely. So, for example, if I have, using an isolator, a lot of people think of the isolator as your primary containment, but actually in a lot of ways, that ends up being secondary or even sometimes tertiary containment. The most ideal, highest potent compounds, you would like to use a closed process inside your environment. So maybe you have, instead of cutting open that bag and dumping it into the tank, where you have an open lid on the tank, maybe you have some sort of transfer valve or some sort of closed system where you can keep that bag closed and do the transfer in a much more closed manner. Now what you have is your primary containment is the bag of your powder with maybe the special valve or connector on it. And then you have your major tank where that has a special valve or connector on it where you can do that transfer, and that's your primary containment. And now your isolator becomes your secondary containment. And then you go to, how hazardous is this? Do I need to have that operation run in, say, negative pressure? And then do I do this inside of a clean room that has a certain level of cleanliness or monitoring in it? So I have a third level of containment and monitoring for my process to make sure that if that's a really nasty thing, of the nastiest, then that never gets out of that primary or secondary containment.
So we're talking about a bag inside a box, inside another box,
Exactly.
just to be clear.
Yep. And then inside that box, inside the third box, which is the room, there are humans typically running around. How do you protect those people in there? And if you're using, if you have a low band substance and you're using already secondary containment, then maybe that's all you need, and you can just use standard gowning that you would use in an ISO 8 type environment or grade C clean room. If you need to go to the next level of, not typically for asepsis, but the next level of safety, then maybe you still do the, personal air protection or some sort of protection by touch for the operators as well.
So who does the swab samples? I mean, it sounds to me like that's something that's got to be done in a very controlled environment. In other words, cleaning samples, yeah, I get that part. But if we're going to do a cleaning swab sample for a potent compound, what kind of approach does your client take there? That's got to be an effort into itself. You know?
It is, and that's the difficulty and the challenge of potent compounds and aseptic processing. Great example, touch plates. So in aseptic processing, you typically will, at the end of every process, you're going to want to do some sort of surface monitoring. Okay? Also, during the process, you've done some viable air sampling, and you have now a whole bunch of auger plates, whether they be Rodak plates or regular auger plates and dishes, and now you need to take those over to the lab. Well, you can't just take them out of the isolator and take them over to the lab. They've had potent compound impinged on them. So how do you contain those properly? So typically what we would do was we would have a bag out process where you would have, say, like a Whirlpack type bag. You would drop each individual plate inside the Whirlpack bag, close it up, wipe it down, put it in a secondary bag, transfer that out of the isolator after you've cleaned everything, and then you can actually remove the outer bag as you take it to the lab and you incubate it in the inner bag. So now, instead of having the naked plate in there, you have something that's contained in a sterile bag, and it provides some challenges to reading, and you can't, I do the IDs as well and all that type of stuff, but again, safety first. You can at least get counts on your plates. Hopefully, if you're dealing with isolation technology and doing aseptic processing, all your plates have zeros, organisms on them anyhow, and it's no issue.
What about amounts of residuals? The same thing, right? You swab the areas and you go through the same triple bagging?
Exactly, and then once you have to have a lot of these same methodologies that you're using for containing the product in manufacturing, you need to have some of those, whether that be the full level or maybe some sort of containment light inside your lab area, because you have to protect your lab personnel as well.
So on a question on that, checking for cleanliness that work, lack of drug substance presence. I mean, cleanliness is almost a foregone conclusion in this case. Okay. What the interesting conversation we had with Craig Johnson this morning from Walker was, well, how do you clean it? And we got into this discussion where it's a wipe down. So I was a little, I thought perhaps some sort of chemical neutralization process or so like a CIP kind of setup, but apparently the wipe down is a lot more contained, a lot more controllable. Would seem to be a lot more variable because it's it's operator dependent. So how do you clean the inside of an isolator?
Good question, and there isn't one answer for that. It really depends on different processes and what, again, how you need to clean it depends on the solubility of your substance and things like that. But the solutions vary from, I have seen, but not in the last 10 years, full CIP wash down, almost like treating an isolator like it's a tank, okay? Typically, though, it's done with just still water, so it would be a rinse in place versus a full cleaning with like a CIP solution or something like that. But then you introduce all these other issues of now you have all this moisture around, and you got to get it dry, and you got to get it, clean.
And you got to get rid of the water.
You got to get rid of the water. So now you have to have drain systems. You have to have contained drain systems. Those need to be emptied out. And now you have, hazardous waste that you need to deal with, which is not inexpensive to dispose of, especially when it's in the liquid phase. It's very heavy, and if you have a containment drain system that's built in your subfloor and things like that, that's very difficult to work with. Okay? So the best systems, the most modern systems I've seen, tend to keep everything inside the zone of the isolator chamber itself. At SKAN and other isolator companies, what they do is typically provide a second set of filters that are in the return air ducts. So before you actually get the air to the return air duct, you've actually HEPA filtered it yet again with a safe change type filter. This way, you don't ever have to clean the return air ducts, because the assumption is, assuming it's a particulate, it'll get captured in that filter, and you'd have sufficient filtration there to keep the ducts clean. And then the inside of the isolator itself, you would do some sort of either wipe down or if you want to do some sort of wet wipe, a rinse in place with a misting wand. So it's much more about misting and getting coverage on there to be able to have something that'll grab onto the substance and allow you to clean it better than it is about actually forcefully rinsing it off with like a spray ball type of approach.
But I gotta believe all of this is contingent upon the chemical properties of the compound we're talking about.
It absolutely is, and again, not only chemical properties, but biological properties of it as well. As it turns out, some of the things that people fear the most, like live viruses and things like that, are some of the easiest things to inactivate, especially in an isolator system that has a biodecontamination or sterilization process built in, like hydrogen peroxide gas. So not only do you run the gassing process at the beginning to establish your aseptic environment, but then at the end of your environment, after you've done your monitoring, you do all your environmental monitoring, you have to close all that stuff up, you need to get those samples out of there. Now you'll run another hydrogen peroxide gassing process, and that will take care of inactivating your biologic. It also sometimes inactivates a chemical as well. So that's something you can look at, and it's great. What's great about that process is that it's built in, it's already validated, and you know that you're getting at least full coverage in the contained space. Yeah.
And again, it's gotta depend on the molecule. Right?
Always. Everything depends on the molecule, and which kind of gets back to not only the risk assessment from a safety perspective, but also a risk assessment from a GMP perspective. Everything that you do, you want to look at it in the context of what's supposed to be happening with that particular substance.
So it sounds to me like everything seems to be a bit of a balancing act, and it looks like risk assessment is a key component of everything we're going to be able to do. I've been involved with certain companies, and again, on the oral solid side, they make their risk assessments based on solubility, which is not a bad idea, but I mean, it's one way of putting up a barrier.
Right.
With the assumption that anything that's early on is, is like death in a bottle. So yeah, we don't deal with it.
Right.
So, but my question, I guess, is one of the things from this conversation is, so we got to balance containment versus aseptic. So how do you do that?
Again, look at your process and decide what's most important. Let's use an example of like a band 2 type substance. It's or a BSL2 type substance where the safety measures need to be are important, but if the hazard got out of the isolation system, then it would still be reasonably safe for the operators. Then we might run that isolator system under positive pressure, okay? And then we would have something in the control system there that would maybe provide what we call a breach mode. So if we had a major leak in the isolator system, all of a sudden the isolator would go negative. It might break our aseptic part of our problem, but then we at least could go into a safe mode of containment mode. The other thing that we do is we look at different zones of the isolator systems. For example, on a classic design where you would have, say, a vial filling machine. You'd have a washer, tunnel, and filler, okay? Between the depyrogenation tunnel and the filler, you might give yourself a positive pressure zone that is a source where all of the air that gets supplied there goes into the isolator or goes into the depyrogenation of it, or tunnel, excuse me. And what that does is it provides a source there to keep anything that's in the fill zone out of the tunnel, okay? So all your losses then have a supplied air loss. Now you've filled your product and you go down to the far end of the machine and you've completed your capping process. After the capping process, in a normal isolator, you would just go exit a mouse hole. You might have some sort of laminar airflow over there, but basically your pressure cascade would be to keep that all positive and you'd be wasting air into the surrounding clean room. Well, in a containment situation, you would probably want to take that to a negative zone, whether you have your, just after your capping, whether you have a negative sink there, or whether you have maybe an exterior vial washer. So maybe the outside of your vial may need to have a wet rinse there. You would want to have that negative for two reasons. One, if you're introducing water into the system, you obviously don't want that going upstream and potentially contaminating any of your upstream parts of your process in the filling zone. But also, that would be your most negative. So once everything's rinsed, then you know the drug substance is gone from the outside of your vial, and then it can safely exit as if it's normal product coming out.
It sounds like there's a whole host of things that somebody has to consider. So how early on do they, so it sounds to me like people early on in the development phase know they have to do this stuff, but how long does it take them to figure out that what they're gonna need is a process? I mean, when do they come to you?
Good question. I mean, the best and most informed customers typically come to us very early on in clinical phase, and they realize that in phase 1, phase 2 clinical trials that they have something that really needs to be scaled up in a smart sort of way. So how are they going to handle that? And the whole scale-up process needs to be addressed as early on as possible, and obviously safety goes along with that hand in hand. We get a lot of customers that come to us when they're just beginning commercial production, and they have a lot more questions to answer, and they have to sometimes go back a few steps and make sure that they develop those assays for figuring out what the residuals really are, and then determining how they could inactivate the product and then validate the fact that they really did inactivate it.
So does anybody do this with a retrofit? Or, I mean, do you see that happen? Or is it usually it's a you follow what I'm saying?
So there are a number of products out there that are older products that have been manufactured less than modern safety would dictate. And they have been done in open operations, and they come to us and say, you know what, we realize that what we're doing is not enough, or we realize that we've been doing this, say, in an open barrier type situation, and we've had personnel with just the pappers or something like that, and not having the full containment, and they want to retrofit that into a full isolation process, and that takes time and is fairly expensive, but it's important because you have to stop at a certain point and say, you know what, what's more important, my operator safety or keeping my product going. And some products are really critical. I had one product that I was working on in my previous life where the customer made about 3 years worth of the product, and they thought they were good, and then they started working on their new aseptic filling line for that, and all of a sudden they had problems, and they were about to run into a drug shortage problem. Well, again, safety first, right? Well, safety first is fine when you're thinking about the operators, but we also think about the safety of the patients, and this was a life-saving chemotherapeutic drug it was an orphan drug for a very unusual application, so they needed to actually go back to their old manufacturing process, make another 6 months worth of the product to give themselves buffer to get the new system up and running. And they took as many good safety steps and measures they could, but using conventional technologies for doing that in their old process.
Well, thanks, Les. I think we've covered a lot here. I think, in particular, I think one of the answers is that there's a lot more to it than just buying an isolator. I mean, none of it lessens which the education of the client, the education of the end user, the implementation of quality systems within that, and the manner in which they manage their risk. And I would think that your input would be beneficial in that case. I assume that's all part of the service.
It absolutely is. At SKAN, obviously, we are more than just a box builder. We're very much a process company, and we enjoy a lot of these challenges and working side by side with our clients to come up with great solutions.
Well, thanks a lot, Les. And that takes us to the close of this session on potent compounds and their manufacture. Thanks a lot.